Hi guys, I am Mohan, Director of Sudhir NEET Biology Academy. Today I will talk about the digestive system, one of the most important topic in your human physiology. See, whatever I speak and whatever I teach you will be according to the NEET point of view. So kindly be attentive to this class. Okay. Digestive system consists of the alimentary canal and the accessory glands. The alimentary canal and the accessory glands, salivary glands, liver and pancreas. So now let us take one by one. The whole alimentary canal is divided into three parts: mouth, buccal cavity, pharynx and esophagus. Up to this point, this is called as the foregut or stomachial region. And then next is the midgut, which consists of the stomach, intestine up to colon, this portion, midgut or mesenteric region. And last part, a third part is the hindgut or the clitoral region, which consists of colon, these portions, ascending, transverse, descending colon, rectum and anus. This consists of the third region, hindgut. So this is the alimentary canal. The cavity inside the alimentary canal is called the enteric cavity. So the alimentary canal opens in the mouth and ends in the anus, up to which there is an opening called enteric opening. Now let us take up one by one. First, mouth. In this mouth you get buccal cavity and then the salivary glands. Now I will talk only about the mouth, the buccal cavity region. The buccal cavity contains mouth and teeth. So these two are important in the buccal cavity region. Number one, tongue, and number two, the teeth. About the tongue, it is freely movable muscular organ that is made up of muscles. You know what type of muscle is made up of? Smooth muscle or skeletal muscle, you know? This is made up of skeletal muscles, no down. That's important MCQ. And then this tongue is attached to the hyoid bone. The only bone, the hyoid bone, to which the tongue is attached. And then this tongue is attached to the floor of the buccal cavity by a fold called frenulum. Note the point. Frenulum is the fold which attaches the tongue to the buccal cavity. Then on the upper side of the tongue you get papillae. There are four types of papillae. One, valate papillae. Two, fungiform papillae. Three, filiform papillae. And then four, foliate papillae. Number one, valate papillae is at the base of the tongue. The valate papillae is at the base of the tongue. The valate papillae, V-A-L-L-A-T-E, valate papillae is at the base of the tongue. This is the largest papillae. And each papillae has got 100 to 300 taste buds on that. They form an inverted V shape at the base of the tongue. Actually they are 8 to 12 in number. So valate papillae, the largest, and 8 to 12 in number, they get more number of taste buds on that. Number two, fungiform papillae, F-U-N-G-I-F-O-R-M, fungiform papillae at the base of the tongue. They are pink colored. Note the point. They are pink colored, mushroom shaped. Here also you get the taste buds. The third one, filiform papillae, Filiform, filiform papillae. It is present on the surface of the tongue, whole surface of the tongue. It is whitish in color. There is no taste bud. So out of the 4 papillae, you must note down this filiform papillae do not contain any taste bud. The filiform papillae is devoid of the taste buds. Very important, note down. Then number 4, foliate papillae, Foliate, foliate papillae, at the lateral margins of the tongue. Here also taste buds are present. So once again I repeat, 1 valate papillae, 2 fungiform papillae, filiform papillae and foliate papillae. Filiform papillae is devoid of taste buds. Very important. Valate papillae at the base of the tongue, fungiform papillae at the tip of the tongue, filiform papillae on the whole surface of the tongue and foliate papillae on the lateral regions of the tongue. These papillae are covered with epithelium called stratified squamous epithelium. Note down MCQ point. So these papillae are covered with stratified squamous epithelium. And surprisingly, you may know there are some glands in the tongue also. They are called as lingual glands. They secrete watery fluids which contain lingual lipase which act on the triglycerides. And now we pass on to the nerve supply to the tongue, how the tongue moves, how the tongue tastes get the taste, or how the brain knows the taste through the nerves. The nerve supply to the tongue. Number 1, facial nerve. It causes the sensation of taste. So facial nerve causes the sensation of taste. Number 2, glossopharyngeal nerve. Number 3, trigeminal nerve, which carries sensation of pain, pressure and temperature of the tongue. Number 4 is the vagus nerve. It is the largest nerve, 10th cranial nerve. That gives the sensation to the back of the tongue. By this vagus nerve, the back side of the tongue get the sensation. Number 5, hypoglossal nerve for tongue movement. So hypoglossal nerve gives the movement for the tongue. So these are the nerve supply to the tongue. I repeat, facial nerve for sensation of the teeth, glossopharyngeal nerve, trigeminal nerve for pain, pressure and temperature of tongue, vagus nerve for sensation at the back of the tongue, hypoglossal nerve for the tongue movement. So now we see the taste areas of the tongue. Tip of the tongue, you know, the sweet taste. Tip of the tongue, sweet taste. Tip and sides, you get the saltish taste. Sides of the back and or sides of the base of the tongue, sour taste. At the base of the tongue, bitter taste. So I repeat, tip of the tongue, sweet. Tip and sides, salt. Sides at the base, sour. And at the base, bitter taste. So these are all about the tongue. Now we come to the next teeth present in the buccal cavity. When we are talking about the human teeth, that is 1 thecodont, then 2 diphyodont and 3 heterodont. So we will see one by one. So what is thecodont? Our teeth are embedded in the socket of jawbone. So they are called as thecodont. Diphyodont. That means in our lifespan, there are two sets of teeth appear. That is, one milk teeth or temporary teeth. Number two, permanent teeth or adult teeth. Actually, the milk teeth begins at the age of 6 to 11 months, completed at the age of 2, out of the teeth, central incisors are the first one to erupt. So first tooth that erupts is the central incisor. The number of milk teeth will be 20. So note down, it is important for NEET point of view. There are 20 milk teeth. They are seen in the baby or in the children. In children, the premolars are absent. Very, very important. Note down. It's also important for the NEET point of view. So I repeat, the milk teeth begins at the age of 6 to 11 months, completed at the age of 2. First one to erupt is central incisor. Total number of milk teeth 20. So the total number of milk teeth in children are 20. There is no premolars in the children. Very, very important. Coming to permanent teeth, it appears between the age of 6 and 12. The last molars come after the 18 years. These last molar cells or teeth are called as the wisdom teeth. Very, very important. Note down. The question has been asked even 2 years back in the NEET question paper. The wisdom teeth constitutes the last 4 molar teeth. Very, very important. So if we take the number of teeth, there are 20 milk teeth and 32 permanent teeth and 28 in teenagers. We will see now. Third, heterodont, that means the teeth differ in the general appearances. There are many types of teeth in our mouth. 4 types of teeth. One, incisors, canines, premolars and molars. I repeat, incisors, canines, premolars and molars. So ICPM. Dental formula, I will tell you the dental formula for the adult, for the children and for the teenagers. For the adult, the dental formula is 2123 by 2123 into 2. ICPM M 2123. The same set below 2123 into 2 sets, 32 teeth. In children or baby, the dental formula will be 2102 2102 into 2. 2102 by 2102 into 2. 0 represents the premolars. So there is no premolar in the children. In the teenagers up to the age of 18 years, the dental formula will be 2122 by 2122 into 228. If you see the dental formula for teenagers, the last molars, instead of 3, there will be only 2 by 2. So the last 4 molars appear after the 18 years as the wisdom teeth. I once again repeat, the wisdom teeth are the last 4 molars. Very, very important for the NEET point of view. So the number of teeth, if you calculate, in children 20, in teenagers 28, and in adult 32. So these are the 3 types of teeth. That is 1, thecodont, 2, diphyodont, and 3, heterodont. Coming to the structure of the teeth, every tooth consists of 4 structures: outer enamel, then inner dentine, cementum and innermost pulp tissue. The portion of the tooth exposed to the oral cavity is known as the crown. The portion below the dental crown is known as the root of the tooth. So I told you the enamel is the outermost part of the tooth, at the top of the tooth. No doubt it is the hardest and toughest part of our body. I will ask one question. It has been asked in the NEET. So enamel actually originates from ectoderm or mesoderm and endoderm. Can you tell me? So in the embryology, the zygote develops into a blastula, and then blastula into a gastrula, and gastrula contains three layers: ectoderm, mesoderm and endoderm, from which all the organs are organized. That is called organogenesis. So all our human parts are developed from these ectoderm, endoderm and mesoderm. Now I ask, the enamel part of the teeth is developed from which portion, ectoderm or mesoderm and endoderm? Can you tell me? See, I'll answer, it is ectodermal in origin. Very important, note down. And then dentine, the inner side of the teeth, consists of calcified connective tissue. So these are all about the tongue and teeth that present in our buccal cavity. Next slide, we will pass on to the next stage, that is esophagus, pharynx and stomach. Thank you. Now let us move on to the next part of the alimentary canal, the pharynx. Pharynx is the common passage for food and air. So actually the esophagus, the food pipe and the trachea, the windpipe, open into the pharynx. Epiglottis is a cartilaginous flap that prevents the entry of food into the glottis. So you must differentiate glottis and epiglottis. So glottis is the opening of the windpipe, trachea. When food is passed on into the stomach or into the pharynx region, this epiglottis prevents the entry of food into the glottis. This pharynx has got three parts. The upper part is called nasopharynx, middle oropharynx, and the innermost or lowermost laryngopharynx. Nasopharynx, oropharynx and laryngopharynx. Uppermost nasopharynx, middle oropharynx and the lowermost laryngopharynx. The lymphatic tissue of the pharynx and the oral cavity are arranged in a ring-like manner called Waldeyer's ring. Note down, it's very important. Waldeyer's ring is the ring formed by the lymphatic tissue of the pharynx and the oral cavity around the pharynx. So note down, Waldeyer's ring, very important for the MCQ point of view. The next comes the esophagus. In the esophagus there is no digestion, no absorption. It does one main function, peristalsis, that is the movement of the food into the stomach. From the buccal cavity, the food is moved into the stomach by the peristaltic movement of the esophagus. Next part of the digestive system is the stomach. Stomach is the J-shaped structure. It also has three parts. The cardiac part nearing to the heart, number 2 fundus, and then pyloric part. So cardiac part is the uppermost part of the stomach, fundus is the middle and body, and finally the last part is the pyloric part. The cardiac part is near the heart and the pyloric part actually opens into the duodenum. Fundus is the middle part wherein the food is stored there for 4 hours. It's also MCQ question, important for NEET. So food is stored in the fundus region of the stomach for 4 hours. So you can easily remember fundus, F first letter, 4 first letter F. So the food remains in the fundus for 4 hours. There are 2 sphincters that actually regulates the movement of food. Cardiac sphincter or gastroesophageal regulates the opening of esophagus into the stomach. Likewise, the pyloric sphincter guards the opening between stomach and duodenum. So these are the parts of the stomach. And the next important point you must note down is the cells present in the stomach. The gastric glands and the cells present in the stomach. Actually, there are 5 types of cells in our stomach, out of which 3 are very important. Let us take one by one. One, the oxyntic cells or parietal cells, other name parietal cells. They are present on the side walls of the gastric glands. There are many oxyntic cells. The main function is that they secrete the HCl in our stomach. Very important. HCl is secreted by the oxyntic cell. It is also an MCQ question for NEET. The number 2, chief cells. They are also called as peptic cells or zygogenic cells. They secrete gastric amylase and lipase. Number 3, mucous cells. They are present throughout the epithelium of the stomach. They secrete mucus. This mucus prevents the stomach from the acidity of the HCl. This makes the gastric juice acidic, actually 1.5 to 2.5. Fourth cell, argentaffin cell, ARGENTAFFIN. Argentaffin cells, they are endocrine glands, actually secrete serotonin, somatostatin and histamine. So very important. These cells secrete serotonin, somatostatin and histamine. And finally, the Gastrin cells. They are also endocrine cells present in the pyloric region. They secrete gastrin hormone. So I repeat, the first 3 are the exocrine glands. Oxyntic cells or parietal cells secrete HCl. Chief cells or peptic cells or zymogenic cells secrete gastric amylase and lipase. Mucous cells secrete mucus. These 3 are the exocrine glands. Argentaffin cells secrete serotonin, somatostatin and histamine. Gastrin cells secrete gastrin hormone. These cells are present in the pyloric region of the stomach. So these 2 are endocrine glands present in the stomach. So these 5 things you must remember, because there is always a question in this area, the cells present in the stomach. So now let us take the next part of the alimentary canal, small intestine. Now let us start the next part of the alimentary canal, small intestine. Actually it is 2.5 centimeter in dia and 3 meters long. It's very important. Now let us move on to the next part of the alimentary canal, small intestine. It is 2.5 centimeter in dia and 3 meters long. Note down this point. And small intestine has also got 3 parts. One, duodenum, jejunum and ileum. Duodenum is C-shaped, jejunum is U-shaped and ileum is coiled, 2 meters long. Then the small intestine opens into the large intestine. The large intestine is 6.5 centimeter in dia and 1.5 meters long. Note down. It has also got 3 parts, cecum, colon and rectum. So cecum has symbiotic microorganisms inside that. It's also called as the appendix. It arises from the large intestine. It's also called a vestigial organ. So appendix is a vestigial organ. Then colon has got 4 parts: ascending colon, transverse colon, descending colon and sigmoid colon. This colon actually opens into the rectum and the rectum opens into the anus. This completes the pathway of the alimentary canal. The intestinal juice, the small intestinal juice, is slightly alkaline. The pH of the intestinal juice is 7.5. You must here also note the cells of the small intestine, which are very important in the role of digestion. There are 3 types of cells in small intestine. 1. S cells. They secrete secretin. It simulates the secretion of pancreatic juice, which is rich in bicarbonate ions. So the small intestine has got a type of cell, S cell, that secretes the enzyme secretin that stimulates the secretion of pancreatic juice that is rich in bicarbonate ions. Another type of cell, CCK cells, that secretes CCK hormone, cholecystokinin hormone, that causes the contraction of the gallbladder to release the bile juice. This is very important. The CCK cells secrete the CCK hormone, cholecystokinin hormone, that causes the contraction of gallbladder and release the bile juice. And finally, it has got another type of cell, K cells. Then, the types of glands present in the small intestine is also very important for the NEET point of view. Number 1, Brunner's gland. Brunner's gland, otherwise called as the submucosal glands. They are present in the duodenum. They secrete enzymes and mucus. They prevent the duodenal wall from the acidity. So question may be asked in the NEET. So the prevention of duodenal wall from acidity is prevented by the enzyme secreted by the Brunner's gland or submucosal glands. Number 2, Peyer's patches. These glands are present in the ileum. They secrete lymph. They destroy harmful bacteria. They are immunological in function. Peyer's patches in ileum. The number 3, crypts of liberculum, formed by the mucosal layer of the small intestine. They are present throughout the intestine between the villi. So crypts of liberculum present in the intestine between the villi. So it has got Paneth cells. They secrete lysozyme. The Paneth cells secrete lysozyme and they act the function of phagocytosis. They do the function of phagocytosis. They engulf the microorganisms. Number 2, argentifin cells. They secrete hormones. They stimulate the smooth muscle contraction. So these are the 3 glands you must know. Brunner's gland in duodenum, secreting mucus, preventing duodenal wall from acidity. Peyer's patches in ileum, secretes lymph, destroy harmful bacteria. Crypts of liberculum throughout the villi, the small intestine. They got 2 types of cells. Paneth cells secrete lysozyme, they do phagocytosis. Argentifin cells, they secrete hormones, stimulates the smooth muscle contraction. And the valves or the sphincters present in the alimentary canal. Number 1, cardiac sphincter between esophagus and stomach. allows food flow into stomach. Pyloric sphincter between stomach and duodenum allows food flow into the duodenum. Ileocecal wall between ileum and the large intestine, food flow into the large intestine. Sphincter of anti-internus at the terminal end of the alimentary canal allows the fecal matter through the anus. So I have separated the sphincters. One cardiac sphincter between esophagus and stomach, pyloric sphincter between stomach and duodenum, ileocecal wall between ileum and large intestine, sphincter of anti-internus, very important, terminal end of alimentary canal allows the fecal matter through the anus. If we take the layers of the alimentary canal, it has got 4 layers. One serosa, muscularis, submucosa and mucosa. First layer, outer layer, the serosa formed by mesothelium and connective tissue. Second layer, the muscularis, formed by the smooth muscles, outer longitudinal smooth muscle and inner circular smooth muscles. So note down another important MCQ for NEET point of view. The muscularis is made up of smooth muscles. The outer smooth muscles are longitudinal and inner smooth muscles are circular. 3 submucosa formed by connective tissue. And lastly, mucosa layer, the inner layer, has got folds and glands. In the stomach region, the mucosa has the fold called rugae. In intestine, the fold of mucosa forms the villi. So you must differentiate between rugae and villi, very important. The mucosa layer of the alimentary canal folds in the stomach as rugae and in intestine as villi. The glands, the mucosa glands in stomach are gastric glands and the mucosa glands in intestine are crypts of liberculum. You must also differentiate these glands of mucosa. Mucosa glands in stomach are gastric glands. The mucosa glands in intestine are crypts of liberculum. Then we pass on to villi, the finger-like projections of mucosa of the small intestine. The villi forms microvilli, brush-like structures which increase the area of absorption. The villi is supplied with a network of capillaries and a large lymph vessel called lacteal, very important. In the lacteal lymph vessel, the fat molecules are absorbed. Method of absorption: absorbed nutrients enter into the capillary to enter blood or into the lacteal for lymph. So the nutrients after absorption through capillary, they enter into the blood or they enter into the lacteal. From the lacteal they enter into the lymph system and they combine with the blood circular system. So the lacteal blood vessel or the lacteal lymph vessel is the most important vessel in villi, which plays an important role in the absorption of fat. So the next video or the next part, we will see the digestive glands separately in depth. Thank you. Now we shall move on to the accessory digestive glands. The glands are the salivary glands, the liver and pancreas. First, let us take up the salivary glands which are present in the mouth. There are 3 pairs of salivary glands. So you must kindly remember that these salivary glands are present outside the buccal cavity, but inside the mouth, but outside the buccal cavity. This is another MCQ. Right. There are 3 pairs of salivary glands: parotid, submaxillary or submandibular, and sublingual. 3 pairs of salivary glands. Parotid gland is the largest gland, present below the cheek. The duct goes out of the parotid is Stenson's duct. It secretes serous fluid and salivary amylase. Second one is the submaxillary or submandibular. These glands are present at the angles of the lower jaw. The ducts coming out of the submaxillary gland is the Wharton's duct. They secrete mucus and enzymes. The third one is the sublingual. It is the smallest gland. It is present below the tongue, sublingual. The duct coming out of the sublingual gland is the duct of Rivinus. It secretes mucus. So here you must definitely in this area you will get a question in the NEET. Parotid is the largest gland and sublingual is the smallest gland. You must remember. Where they are present, parotid below the cheek, submaxillary or submandibular at the angle of the lower jaw, and sublingual, the smallest gland below the tongue. Another thing you must note here is that the ducts coming out of these salivary glands, very, very important. Definitely one question will come in the NEET. Stenson's duct comes out of the parotid, Wharton's duct from submaxillary, and duct of Rivinus from sublingual. So this is about the salivary glands. Next we come to the liver. The liver is the largest gland present in the human body. It weighs about 1.2 to 1.5 kg. Of the total weight, 1/40th is the weight of the liver. It has got 2 lobes and each lobe is further divided into lobules. The lobules are the structural and functional unit of the liver. Each lobule of the liver is covered by Glissen's capsule. Very important, note down a question from NEET. Each lobule is covered by Glissen's capsule. So Glissen's capsule is present in the liver. This is made up of what type of tissue? Can you tell me? This Glissen's capsule of the liver is made up of what type of tissue? This is a connective tissue sheath. You must remember. You must note down. Then the Kupffer cells present in the liver act as the phagocytes. They engulf the microorganisms. They eat the dead cells and the bacteria by phagocytosis. Kupffer cells. Then another important thing you must know is that the heparin, the chemical, is an anticoagulant produced by the liver. Another area of NEET question may come. So the heparin is the anticoagulant produced by the liver. Liver produces the anticoagulant heparin. So you may wonder that how the blood is fluid inside our blood vessels. While the blood comes out, it actually clots. How does it in a liquid condition inside the blood vessel? It is because of this heparin. That heparin is produced by the liver. And then liver produces prothrombin and fibrinogen. Prothrombin and fibrinogen. These two proteins help in the blood clot. These things we will see in the chapter on the circulation, how the blood clots, etc. So the prothrombin and fibrinogen, these proteins are very helpful, are the basic proteins for blood clot. These proteins are produced by the liver. In this area, I want to mention all the functions of the liver so that you may get the overall idea of the liver. You can answer any type of question. The function of liver, number 1, glycogenesis. So what is glycogenesis? The formation of glycogen from sugar. The excess of sugar in our body is converted into glycogen. That is called glycogenesis. That is done by the liver. Glycogenolysis, just opposite. That is a breakdown of glycogen into glucose. Third function, gluconeogenesis. Formation of glucose from non-carbohydratic substrates. Actually, the glucose is formed from the carbohydrate. The simplest form of carbohydrate is this glucose. The gluconeogenesis is the formation of glucose from non-carbohydrate substrates. Then number 4, deamination. Number 5, the liver takes place in the ornithine cycle. The formation of ammonia when you study the excretory system, we will see. The formation of ammonia is due to the liver forming the ornithine cycle. Sixth function, detoxification of drug. So even if we take alcohol or any other drug, that goes to the liver and then the liver detoxifies the drug. Seventh function, it stores the fat and stores the fat soluble vitamins A, D, E, K. So you know the vitamins, fat soluble and water soluble. B and C are water soluble. A, D, E and K are fat soluble vitamins. Liver stores the fat and the fat soluble vitamins. It also synthesizes vitamin A. So this is all about the liver and the overall functions of the liver. Then third gland we come to, before that, we take up gallbladder. The gallbladder is present in the liver. The gallbladder actually stores the bile. The function of gallbladder, so you must distinguish here. Bile is secreted by the liver but stored in the gallbladder. Gallbladder doesn't secrete bile, but it is only a storage portion of the bile. You must differentiate. You must remember. It's also MCQ. So gallbladder acts as a storehouse of the bile secreted by the liver. Now coming to the bile, the function of bile, it makes the food alkaline with a pH of 7.5 to 8.6. So you know the pH scale, 1 to 6.9 acidic. So 1 is the most acidic, 6.9 less acidic, 7 neutral, 7.1 to 12 is the basic. So here the bile makes the food acidic with a pH of 7.5 to 8.6. Bile has got no enzyme. Very, very important. Very important. Bile contains no enzymes, but it helps in the emulsification of fat. How? It contains bile salts 6%, water 92%, and bile pigments 0.3%. So what are the bile pigments? Bilirubin and bilirubinin. Bilirubin is yellow in color, whereas bilirubinin is green in color. It contains fatty acid, cholesterol, phospholipids and lecithin. So I repeat, the bile contains water 92%, bile salts, bile pigments, bilirubin yellow in color, bilirubinin green in color, note the point, fatty acid, cholesterol, phospholipid and lecithin. So what is the function of lecithin here? The main function of lecithin is that it decreases the surface tension and helps in fat emulsification. So the fat is emulsified by the decreased surface tension of the lecithin present in the bile. Now next we come to pancreas. It's an elongated leaf-like structure present between the limbs of C-shaped duodenum. Pancreas is both exocrine and endocrine. So it's also very important. It's another area where you get a question often. Which is a gland both exocrine and endocrine? Pancreas. How? It secretes pancreatic juice and acts as exocrine in function. And it secretes insulin and glucagon and acts as the endocrine gland. So actually pancreas secretes the pancreatic juice. So it is the exocrine mode. Endocrine means it has got alpha and beta cells. Beta cells of pancreas secrete insulin, alpha cells secrete glucagon. It's also, you must remember, beta and alpha cells. Beta cells secrete insulin, alpha cells secrete glucagon. The pancreatic juice is alkaline, pH is 7.5 to 8 due to the presence of sodium bicarbonate, another area of NEET question. So how this pancreatic juice is alkaline? How the pH comes to 7.5 to 8? Because the pancreatic juice contains sodium bicarbonate. So now we have to see the biliary system, the duct formation, how the bile is secreted into the duodenum, how the pancreatic juice is secreted into the duodenum. Let us see now. From the liver, left and right, two ducts arise and they combine to form a common hepatic duct. This common hepatic duct joins with the duct called the cystic duct from gallbladder and forms a common bile duct. This common bile duct combines with the pancreatic duct to form the hepatopancreatic duct. So this hepatopancreatic duct opens into the duodenum and releases the pancreatic juice and bile into the duodenum for absorption or for digestion. So it's very important. Biliary system is formed by the duct system of liver, gallbladder and pancreas. So I repeat, carefully listen. Two ducts from the liver combine to form a common hepatic duct. This hepatic duct joins with the cystic duct of gallbladder to form the common bile duct, and this common bile duct combines with the pancreatic duct to form the hepatopancreatic duct. It opens into the duodenum, releasing the pancreatic juice and bile. Pancreatic juice from pancreas and bile from the liver. So the cystic duct is from gallbladder. You must know. Hepatic duct from liver. Hepatic duct from liver plus cystic duct from gallbladder forms common bile duct. These are the areas where you get questions. But very careful. This common bile duct plus the pancreatic duct forms the hepatopancreatic duct, which opens into the duodenum. So these are all about the structure we find in the digestive system, alimentary canal and the accessory digestive glands. The next coming video, let us see the mechanism of digestion. Now we shall move on to the mechanism of digestion. So digestion is a process of converting macromolecules into micromolecules, that is carbohydrates into glucose, proteins into amino acids, fats into fatty acids and glycerol. So digestion takes place in two forms, one mechanical in mouth and chemical, that is enzymatic, in stomach and small intestine. First, let us take up the digestion in mouth. In mouth, mastication takes place, that is the biting and grinding of food, chewing of food. This chewing of food is controlled by the somatic nerves, another area you can get the question. So the chewing of food in the mouth is controlled by the somatic nerves. Number two, deglutition. It is the process of swallowing and swallowing the masticated food. Saliva secreted by the salivary glands help in lubricating and adhering the masticated food into small ball called bolus. It passes through the esophagus to the stomach by peristalsis movement. When we see the secretions of the mouth, saliva is the most important one here. Saliva. The functions of saliva: it softens the food, it helps in tasting the food, it kills bacteria, and it digests starch into maltose. The saliva secreted by the salivary glands has the pH 6.8. What are the constituents of saliva? Another area I get the question. The constituents of saliva: sodium, potassium, chlorine, bicarbonate. These electrolytes: sodium, potassium, chlorine, bicarbonate. The antibody IgA, the enzyme salivary amylase or ptyalin, and lysozyme. This lysozyme kills bacteria and prevents infection. It's antibacterial in action. The enzymatic action of saliva: the saliva secreted by the salivary gland in the mouth digests starch into maltose. The pH of salivary amylase is 6.8. So actually 30% of the starch is converted into maltose in the mouth. Second stage of digestion in stomach. The food mixes thoroughly with the acidic gastric juice by the churning movements of its muscular wall to form a structure of chyme. See, what are the chemicals secreted in the stomach? Number one, HCl, secreted by the oxyntic cells of the gastric gland. It activates the inactive pepsinogen into pepsin. It makes the food acidic. pH becomes 1.8. So this acidic medium is favorable for the protein digestion of pepsin. If there is no HCl in the stomach, the pepsin doesn't work because it acts only in the acidic medium. The pepsin secreted by the chief cells of the gastric cells converts protein into proteases and peptones. Third one, the chemical renin secreted by the chief cells of the gastric glands. It digests milk protein. It acts on soluble milk proteins, casinogen, and converts into insoluble casein in the presence of calcium ions. So here, for the action of renin, calcium ions are very important. Casein is precipitated as insoluble casein-calcium compound called curd. So the caseinogen with calcium and renin becomes casein, and finally it is converted into curd. Fourth one, gastric lipase. It converts fats into fatty acid and glycerol. A small amount of fat is converted. And mucus and bicarbonates in the stomach helps in the lubrication. Then we come to the digestion in the small intestine. So in the small intestine, the duodenum part, actually this bile from liver and the pancreatic juice from the pancreas come through the hepatopancreatic duct, and the intestinal juices also join. So here three juices: bile from liver, pancreatic juice from pancreas, and the intestinal juices, Saccus entericus, from the small intestine. Now let us see the action of bile. The pH of bile is 6 to 6.7. I already told you there is no enzyme in the bile. By the bile salts, it makes the fat emulsified. So it helps in the emulsification of fats. Then number 2, let us take up the enzymes of the pancreatic juice. Its pH is 7 to 8. The trypsinogen, it is inactive. This inactive trypsinogen is converted into trypsin by the enterokinase secreted by the intestinal juice. So you must know this point. The trypsinogen is inactive. This inactive trypsinogen is made active by the enzyme enterokinase that is secreted by the intestinal mucosa. This trypsin acts on proteins and converts into polypeptides and peptones. Chymotrypsinogen converts protein into large peptides. Procarboxypepidases converts polypeptides into dipeptides and amino acid. Pancreatic amylase converts starch into maltose. Pancreatic lipase converts triglycerides into fatty acids and glycerol. Then enzymes of intestinal juice, maltase, so the pH is 7.6. The maltase is converted by the maltose into glucose. The enzyme sucrase converts sucrose into fructose and glucose. The enzyme lactase converts lactose into galactose and glucose. Dipeptidases converts di and tripeptidase into amino acids. Nucleotidase converts nucleotides into nucleosides and phosphoric acid. Nucleosidase converts nucleosides into base and sugar. Lipases converts fats into fatty acids. So you must note here, gastric and intestinal juices are stimulated by neural signals. Very, very important. The neural signals coming from the brain control the gastric and intestinal juices. Right, now carbohydrate is digested into glucose, protein into amino acid, and fat into fatty acids. They are ready for absorption. Now we come to the mechanism of absorption in the small intestine. It takes place by 3 ways. One, active method. Number 2, by simple diffusion. Number 3, facilitated transport, passive, with the help of some carrier proteins. So glucose, so here is the area, another area where you can get a question from the NEET. Glucose, amino acids and sodium are absorbed by active transport. Some amount of glucose, some amount of amino acids and chloride ions are absorbed by simple diffusion, and some amount of glucose and some amount of amino acids are also absorbed by passive movement. The absorption of fatty acids and glycerol is a separate way. These fatty acids are made into small droplets called micelles, and these fat globules are transformed into chylomicrons. These chylomicrons are transported into lymphatic vessel, the lacteals in the villi, and from this lacteals through the lymph, the chylomicrons are open into the bloodstream. So here the fatty acids are absorbed by the lacteals. So very important. Now I will give a short idea about the different absorption in different parts of the digestive system. In mouth, some drugs are absorbed. In the stomach, water, glucose and alcohol are absorbed. In small intestine, glucose, fructose, fatty acids, amino acids and glycerol are absorbed. In the large intestine, water is the most important thing, is absorbed, and some minerals are absorbed. So I repeat, some drugs are absorbed in the mouth, water, glucose and alcohol are absorbed in the stomach, glucose, fructose, fatty acids, amino acids and glycerol are absorbed in small intestine, water and some minerals are absorbed in the large intestines. Now finally we come to the disorders of the digestive system. One, jaundice, that is the dysfunction of the liver, deposit of bile pigments. Hepatitis, inflammation of liver. Diarrhea, increased fluidity of fecal matter. Constipation, fecus retained in the colon, indigestion. Alactasia, that is the lack of enzyme needed to digest milk sugar. Alactasia is the disease in which there is an enzyme is lacking needed to digest the milk sugar. Peptic ulcer, two types, duodenal peptic ulcer in duodenum and gastric peptic ulcer in stomach. Eroded area of the tissue. What do you mean by the ulcer? Actually, the lining of the stomach and duodenum are eroded. It is actually caused by the bacterium Helicobacter pylori. Helicobacter pylori. Another disease, hernia. These are the ruptures in the abdomen. There are three types of hernia: inguinal hernia, femoral hernia, umbilical hernia. Inguinal hernia is the rupture through the inguinal canal. Femoral hernia is the hernia in the part of groin. Umbilical hernia in the babies behind the navel part. The most complicated hernia is the strangulated hernia, most dangerous, wherein a section of the intestine becomes twisted and get trapped in the bulge. So blood supply is being cut off and severe pain occurs. For this, immediate surgery is the only remedy. Then another disease is the inflammation of gallbladder, cholelithiasis. That is the inflammation of the gallbladder. Then another thing, protein energy malnutrition, PEM, in children. So these two, Marasmus and Kwashiorkor, are the two diseases of children of protein malnutrition, PEM. So now I will give an idea of the Marasmus and Kwashiorkor. Similarities of Marasmus and Kwashiorkor I will just list out. In both the Marasmus and Kwashiorkor, thinning of limbs is there, and then the growth and body weight decline, growth and brain development are impaired, and wastage of muscles. So these are the things common to Marasmus and Kwashiorkor. I repeat: thinning of limbs, growth and body weight declines, growth of brain development impaired, wastage of muscles present. So how will you differentiate? Marasmus occurs lesser than 1 year. Kwashiorkor mostly occurs in more than 1 year babies. Protein calorific value low. So both protein deficiency and low calorific value in Marasmus, whereas in Kwashiorkor, low protein and high calorific value. In Marasmus, no edema of belly and leg is seen, whereas in Kwashiorkor, there is an edema of belly and leg. Swelling of the body, not present in Marasmus. The swelling of body you can see in Kwashiorkor. In Marasmus, appetite is good. Kwashiorkor, poor appetite. Occurrence, Marasmus more common, Kwashiorkor less common. Liver enlargement you cannot see in Marasmus, whereas the liver enlargement is present in the Kwashiorkor. The skin is thin and dry in Marasmus, whereas it is not so in the Kwashiorkor. So these are about the protein, energy, malnutrition is also another important area. Then finally we will finish with the calorific value of carbohydrate, protein and fat. The calorific value of carbohydrate 4.1 kilocalories per gram, protein 5.6 kilocalories per gram and fat 9.4 kilocalories per gram. So with this, I hope we are coming to the final stage of this digestive system. Now, if students kindly listen, if you have any queries or if you have any doubts or if you want to write a model test in this chapter on digestive system, just we have begun now, kindly comment through your comment section. I will reply immediately accordingly. So next video will be on the circulatory system. Till then, goodbye. Thank you.